Thanks for tuning in. Please like, subscribe, and check out my Instagram for cool science and not science stuff. Welcome back to Biochemistry on Catalyst University. My name is Kevin Tokoph. Please make sure to like this video and subscribe to my channel for future videos and notifications. In the previous video, we built off of atherosclerosis by talking about advanced glycation end products. If you want more detail on that, go back and watch the previous video. But the advanced glycation end products, as you can see right here, bind to receptors on certain cells. For example, this is a macrophage, an immune cell, and they activate that receptor called RAGE. And that leads to the activation of several pathways, two of which are the NF-kappa B pathway, which leads to the production of inflammatory cytokines, and the NOx pathway. This is NADPH phagosome oxidase, which results in the production of reactive oxidative species, so free radicals. Remember, those free radicals can oxidize LDL, and that, of course, forms, well, oxidized LDL, which we call LDL-ox. That's how we abbreviate it. And LDL in particular, of lipoproteins, is particularly susceptible to oxidation. HDL can be oxidized, but it's not near as susceptible. LDL's very susceptible to oxidation. Okay? And we're going to talk here about how that oxidation occurs and why it's clinically relevant. So here is an LDL particle. Now, LDLs are lipoproteins, right? So the outer portion of them is going to be polar or hydrophilic because it has to be soluble in the blood, and the internal component is going to be where the hydrophobic parts are. Okay? Now, in general, LDL's function is to transport cholesterol to peripheral tissues. And people have this idea, oh, it's this cholesterol that's bad. No, it's not cholesterol that's bad. There's just certain pieces of the LDL that are susceptible to oxidation. In particular, these polyunsaturated fatty acids. Okay? Remember, the outer part of the lipoprotein is actually mostly phospholipid. Out here you have the polar heads, and you have certain proteins here that help increase the solubility in addition to having other functions. Then the internal parts here, in the very center of the core, that's cholesterol, or cholesterol esters. And then here you see in gray, sort of, this is a cross-section, but these are the tails, the hydrophobic parts of the phospholipid. And in the membrane phospholipid tails, you might actually have a polyunsaturated fatty acid. So if you look over here on the right side of the screen, you see an example of a phospholipid that you might actually find in a lipoprotein like LDL. So down here at the bottom, this is the head group. This is actually inositol 4,5-bisphosphate. Here's the connecting phosphate. Here's the glycerol backbone, and then you see two fatty acids. One of them on the right here is saturated, and the other over here on the left is polyunsaturated. Now, if I asked you, is it the polyunsaturated fat on the left or the saturated fat on the right, which one is more of a problem for oxidation by free radicals? What would you say? Well, if you said the saturated fat, you'd actually be wrong. Saturated fats get all sorts of a bum rap in the news and on the media and online. But it's actually the polyunsaturated fat here that plays more of a problem for LDL oxidation. And it's because of these double bonds, they're more susceptible to oxidation by free radicals. Remember that when we have excessive glucose, or if we have anything that's just generally inflammatory that leads to these advanced glycation end products, they lead to the production of reactive oxidative species, free radicals. And those free radicals then oxidize LDL. Let's actually see how that occurs here. So here I'm going to show you a brief mechanism. Here we have a polyunsaturated fatty acid. Here's a hydrogen right there. And we're going to put in a hydroxyl radical. These hydroxyl radicals can abstract these hydrogens. And notice what happens when it abstracts this hydrogen. It leaves at that same position a radical. Now, what this does is it converts this hydroxyl radical into water. But unfortunately, it leaves a radical electron right here. Now, this radical electron will then rearrange with this double bond. Now the double bond's right here and the radical's here. Now, this radical can then couple with molecular oxygen. Molecular oxygen we typically like to write as O2, but it can react with oxygen in this way also. This is another valid structure for oxygen. It itself is a radical. And that oxygen can then couple with this free radical on the lipid, and now we have this oxygen-oxygen group right here, where there's a radical right here. Now, eventually, this oxygen right here with the radical can pick up a hydrogen, and you end up with what's called a peroxy group. But again, this process that I've just shown you is called lipid peroxidation. Okay? Lipid peroxidation typically occurs on polyunsaturated fatty acids, when they're exposed to reactive oxidative species like a hydroxyl radical. And to be fair, this is not the only reaction that can happen, but this is a very common one that occurs with hydroxyl radicals. Now, when you get this lipid peroxidation, there's a few things that can happen. One, you can get radical propagation. Again, notice that if this oxygen right here with the radical doesn't pick up a hydrogen, it still has a radical. And radicals propagate. They spread that radical to another molecule, and so on and so forth. And so you get a vicious cycle of radical propagation. In addition, you're probably also still having the formation of more reactive oxidative species, assuming that you're having these dietary problems in the first place. The other thing that happens is, again, this polyunsaturated fat, remember, is on an LDL, right? This polyunsaturated fat is part of that LDL. It's in the membrane. So by having this peroxidation, you've now oxidized that LDL. And if this happens significantly to that LDL, then you're going to have vascular changes that occur, again, leading to the progression of that atherosclerosis. The other thing that can happen is you can get the formation of these polyunsaturated fatty acid aldehydes. Okay? I'm not going to discuss how this actually occurs because it's a fairly complicated mechanism, but once you get this lipid peroxidation like this, there can be spontaneous, again, non-enzymatic reactions that occur that form aldehydes like this. This compound over here, this is called malondialdehyde. Okay? Malondialdehyde forms when you have oxidative stress in tissues, particularly with lipid peroxidation, and this molecule is very bad. Hopefully this will explain why. So I've got two proteins right here, one protein up here at the top, one at the bottom. And you'll notice that they both have these lysine residues. Lysine is very reactive to things like this, particularly aldehydes. We saw that when lysine reacted with glucose to form the advanced glycation end product in the previous video. But what you'll see here is actually that this nitrogen of the lysine on one protein can chemically react with the aldehyde on one side of this malondialdehyde. And notice what happens. You get this Schiff base formation, or nitrogen-carbon double bond, and now you have this aldehyde, or this carbon component of the aldehyde, physically bonded to this protein. All right? But now I've got this protein here at the bottom, also with a lysine residue, and it can also chemically bond with this other aldehyde here on the bottom of this carbon skeleton, and it forms a Schiff base linkage, or a nitrogen-carbon double bond. You see a problem with this. Not only do we have a protein that's been modified, well, we actually have two proteins that have been modified, and now they're chemically linked. We now have what's called a protein-protein adduct. And this is clinically relevant because, like the AGEs, the advanced glycation end products, these protein-protein adducts can also bind to and activate RAGE, the same receptor that AGEs acted on. And I don't want to go into this in too much detail, but remember, if the protein-protein adduct can activate RAGE, it can also activate these other pathways. It can activate the production of more reactive oxidative species through NOx, NADPH phagosome oxidase. It can also activate the NF-kappa B pathway, which in this case will lead to the production of many inflammatory cytokines. Either way, you've got oxidation, more of it, and you've got more inflammation. And so when we produce these aldehydes from the polyunsaturated fatty acid peroxidation, we get the protein-protein adducts and we get oxidative stress. Oxidative stress we get again through binding of the protein-protein adducts to that receptor through RAGE. Now, one thing you might be thinking, well, there's a huge number of proteins that can become adducted together, right? Well, they've studied that and it turns out it's true. So different adducts of proteins are going to have different binding affinities and different abilities to activate the RAGE receptor. But again, the point is these protein-protein adducts, bad. We don't want those because they cause oxidative stress via binding to RAGE. And how do we get those protein-protein adducts? Well, if we have lots of reactive oxidative species, right? Because we get that lipid peroxidation. How do we get the reactive oxidative species? Well, we have advanced glycation end products. Well, how do we get those? Sugar. So I guess this was a long-winded way of also saying that sugar is really not good for you, especially when it's in high amounts. If you think back 300 years, they wouldn't have added refined sugar to a lot of things. You couldn't just go to the store and buy all sorts of sugary foods. Again, it's not to say sugar in fruits is bad. No one's arguing that. Again, that's natural sugar. We're talking about refined added sugar that tends to be very high in a standard American diet. And that's why we see so many of these health problems, and particularly atherosclerosis in our society. So hopefully this video gave you a good understanding of LDL oxidation and how it's produced by free radicals, and also how it leads to the formation of protein-protein adducts. Please make sure to like this video and subscribe to my channel for future videos and notifications. Thank you.